CRISPR technology has been in the news a lot over the past few years, but what exactly is CRISPR, and can we use it to treat FSHD? CRISPR originally referred to a primitive immune system that evolved in bacteria, but more recently it's been developed as a tool for modifying the genomes of virtually any species. And the beauty of this approach is that when we're talking about therapy, CRISPR has the potential to cure the root cause of a genetic disorder, rather than just treating downstream effects or symptoms. So how does this work? Most genetic diseases are caused by a mutation in a gene that codes for a protein that performs some critical function in the cell. So when you have a mutation in this gene, if a protein is produced at all, it's non-functional and sometimes pathogenic. Now the CRISPR system has two components, a protein called Cas9, which cuts DNA like a pair of scissors, and a strand of RNA which binds to Cas9 and recruits it to any matching sequence in the genome. So Cas9 will cut DNA at any place targeted by the guide RNA. In some cases, you can just cut the original DNA, and the cell's own repair mechanism will fuse the broken ends, and this is enough to solve the problem. But you can also provide a donor piece of DNA, which has the correct sequence, and another repair pathway in the cell will insert the new DNA in place of the old, effectively correcting the gene and allowing the production of a normal, functional protein. But FSHD is different. In FSHD, you have a gene called DUX4, which should be off in skeletal muscle, and instead it's on, and this results in the production of a pathogenic DUX4 protein. The problem is, DUX4 is part of a highly repetitive sequence, and targeting Cas9 to any part of DUX4 will result in multiple breaks in the genome. And this triggers genomic instability and can trigger cell death. But fortunately, there's an alternate system called CRISPR inhibition. Here, Cas9 is mutated, so it's incapable of cutting, and it's fused to a repressor protein. So now the guide RNA recruits a system that represses DNA instead of cutting it. And this is ideally suited to FSHD because we want to turn off or dial down expression of DUX4. And importantly, other repetitive sequences in the genome that match are normally repressed, and so recruiting a repressor to those locations shouldn't cause any problems. Most of the controversy surrounding CRISPR comes from the idea of using it to permanently alter the human genome, especially in ways that can be passed down to our kids. So it's important to point out that what we're talking about here is not modifying eggs or sperm or embryos in any way. We are simply trying to use the system to correct muscle cells in FSHD. And with CRISPR inhibition, we can do this without making any permanent changes to our DNA. The CRISPR is a very powerful and exciting tool, and we're working hard to develop it as a therapy for FSHD.